Within the bodies of those unlucky, a dark force corrupts healthy blood cells with a single touch. The production of haemoglobin weakens and the body struggles to keep up with the damage. Once, a brave hero emerged to fight this relentless foe, HBV 569. Despite its strength, it wasn't enough to defeat beta thalassemia entirely. Until now, enter the mighty BB305, stronger, faster, and more precise. Armed with an optimized design, this hero asserts superior transduction efficiency and enhanced gene expression. Could BB305 be the ultimate solution for beta thalassemia? Welcome to the genetic basis of stuff and things. Let's find out. Scientifically speaking, how could beta thalassemia and other genetic disorders be defeated? The advance of genetics has armed us with the ability to treat diseases by modifying specific genes via gene therapy. Beta thalassemia is a genetic disease caused by mutation of the HBB gene, which leads to reduced beta-globin chain synthesis, or in more severe cases of the disease, absence of beta-globin chain synthesis. There are more than 350 known mutations for beta thalassemia, which can occur on different parts of the gene, such as the promoter, intron, splice sites, and exons. The HBB gene encodes the beta-globin subunit of haemoglobin, an iron-rich protein that transports oxygen in red blood cells. Haemoglobin consists of 2 alpha-globin chains and 2 beta-globin chains. A decline or absence of beta-globin results in excess unpaired alpha-globin, which forms insoluble aggregates that damage the membranes of red blood cells. This causes hemolysis and ineffective erythropoiesis due to apoptosis of red blood cells. This means that patients experience chronic hemolytic anemia, as well as iron overload, as excess iron accumulates in the organs due to the body's inability to eliminate it. Further, the low levels of oxygen cause hypoxia, which leads to enlargement of the bones, liver, and spleen. Living with beta thalassemia requires many treatments to alleviate symptoms, such as regular blood transfusions and iron chelation therapies. Previously, the only curative option was allogenic hematopoietic stem cell transplantation, which is inaccessible to many patients as it requires a human leukocyte antigen-matched donor. Here, autologous gene therapy offers new hope as a curative treatment for patients. Lentiviral vectors have the ability to permanently integrate transgenes into a host cell and allow stable long-term expression of the transgenes. The vectors encapsulate their single-stranded RNA within vector particles, which undergo reverse transcription and form a double-stranded DNA provirus that integrates into the host genome. The HBV 569 lentiviral vector was one of the first used in clinical trials for gene therapy in transfusion-dependent beta thalassemia. The HBV 569 lentiviral vector is built from the backbone of the human immunodeficiency virus and encodes the modified beta-globin transgene, beta-globin T8-7Q, which contains regulatory elements that promote high expression in adult erythroid cells while also maintaining normal hemoglobin function and oxygen carrying capacity. After trialing HBV 569, the results suggested the vector had low titer and modest transduction efficiency, as well as a risk of insertional mutagenesis, where proto-oncogenes can be upregulated and trigger oncogenesis. Modifications were made to improve the vector without compromising safety, creating the BB305 lentiviral vector. The HIV-U3 region in HBV 569 was replaced with a cytomegalovirus promoter to boost transduction efficiency and HBB expression. This meant that the TAP gene and the CHS4 insulators could be removed, making vector production more efficient. During a preclinical trial in 2015, hematopoietic stem cells were transduced with BB305 and then transplanted into mice models of beta thalassemia. During the trial, the BB305 lentiviral vector successfully integrated into the host genome, resulting in sustained production of functional hemoglobin without any adverse effects or insertional mutagenesis. Additionally, the results demonstrated that the lentiglobin BB305 vector could be produced with 3 to 4 times higher functional titers compared to the lentiglobin HBV 569 vector. Clinical trials with the BB305 vector have seen patients significantly reduce transfusion volume and frequency, and even has removed the need for transfusions in some cases. Over the years, various lentiviral vectors have been tested and improved to enhance their efficiency and safety. While the BB305 vector has shown promise, newer technologies such as CRISPR-Cas9 are being explored in clinical trials to find even more effective treatments. The BB305 vector itself has also been modified to better treat those with transfusion-dependent beta thalassemia and improve therapeutic outcomes. As researching and trialing progress, the future of gene therapy for beta thalassemia looks optimistic. The continued evaluation and refinement of these therapies will facilitate the development of a life-changing curative option for those affected by the disease.